A current phenomenon called photosynthesis produces carbohydrates by the reductive condensation of carbon dioxide in the presence of sunlight. Classification of carbohydrates. Carbohydrates can be categorized based on their size, number of the carbon atom, functional group, and complexity. A broad range of classification of carbohydrates based on their complexity is outlined below. 1. Monosaccharide. It comprises the simplest and smallest unit of carbohydrate. 2. Disaccharide. It is made up of two units of monosaccharides joined by a glycosidic bond. 3. Polysaccharide. It is composed of multiple units of monosaccharides connected by glycosidic bonds and exists as polymers. Monosaccharides. The simplest form of carbohydrates is comprised of polyhydroxyaldehyde or ketone that cannot be hydrolyzed further to give simpler sugar. They may again be classified on the basis of the nature of the carbonyl group, i.e., aldose or ketones. These aldose and ketose form can be further categorized based on the number of carbon atoms present in the molecule structure. Table 1.1. 1. Biochemistry. Structure and functions of carbohydrates. Carbohydrates are generally considered as sugar compounds that have the formula CnH2O and or CnH2ON1. Saccharides are crystalline carbohydrates that comprise simple sugars, monosaccharides, and complex sugars, disaccharides, and polysaccharides. Saccharides are sweet in nature and can be found in various forms in natural compounds. Glucose, a monosaccharide, is the simplest sugar and is found in the blood. Sucrose is a common disaccharide comprised of two monosaccharide units, glucose and fructose, and occurs in plants, through which table sugar can be produced. Polysaccharides are composed of long series of monosaccharide units joined together through glycosidic linkage and occurs as cellulose and starch in their natural form. The basic skeleton of the carbohydrates contains 1. Carbon. 2. Hydrogen. 3. Oxygen. Hence, they can also be termed hydrates of carbon based on their empirical formulas. Carbohydrates are usually considered aldoses or ketoses, depending upon the functionality present in the molecule. Molecules possessing an aldehyde functional group are known as aldoses, while with ketone functionality, they are called ketoses. One of the most naturally No. Of carbon. Atoms monosaccharides aldose form ketose form. 3 trioses glyceraldehyde dihydroxyacetone. 4 tetroses erythrose and threose erythrulose. 5 pentoses arabinose, lixose, ribose and xylose. Ribulose and xylulose. No. Of carbon. Atoms monosaccharides aldose form ketose form. 6 hexoses allose, altrose, galactose, glucose, glucose, ketose. Mannose and tallose. Fructose, sucrose, sorbose and tagatose. 7 heptoses menaheptulose, sadoheptulose. 8 octoses 2 keto 3 deoxymanooctanate. 9 nonoses cellulose. Table 1.1 Classification of monosaccharides. The carbon center of monosaccharides having attached hydroxyl groups are often chiral in nature, that produces stereoisomers of the monosaccharides. They can exist in an enantiomeric as well as diastereoisomeric forms due to the presence of multiple asymmetric carbon center. To assign the exact configuration of asymmetric center with regards to a functional group, saccharides are designated with symbol C and L. The difference in configuration of only one asymmetric carbon center of saccharides gives rise to epimers. For example, 6 carbon aldoses such as D-glucose and D-mannose possess different configurations only at the C2 position. Therefore, they are said to be epimers at the C2 carbon center. Likewise, D-ribose and D-xylose are C3 epimers, whereas D-glucose and D-galactose, as well as D-fructose and D-tagatose, are C4 epimeric stereoisomers. Figure 1.1. Figure 1.1. Stereoisomers of hexose sugar molecules. Glucose and mannose differ in C2 configuration and hence are C2 epimers. Likewise, D-ribose and D-xylose are C3 epimers, while D-fructose and D-tagatose are C4 epimers. Structure of monosaccharides. Structural aspects of monosaccharides can be described with the help of the Fischer projection, which explains the open chain structure of saccharides, and the Hayward projection, which explains the cyclic structure of saccharides. Based on several chemical and structural evidence, the monosaccharide can be said to exhibit two types of structural forms. 1. Open chain. 2. Cyclic. Fischer projection depicts the three-dimensional structure of carbohydrates in two dimensions. It involves the projection of all horizontal bonds towards the viewer, while vertical bonds are oriented away from the viewer. Since the rotation of bonds by 90 degrees or 270 degrees in the plane leads to a change in orientation of bonds with respect to each other, which results in the production of enantiomers of the molecule. Therefore, a Fischer projection is ruled out to be rotated by 90 degrees or 270 degrees in the plane. Rotation of bonds through 180 degrees does not cause any change in the molecule's representation, and hence 180 degrees rotation is freely allowed. Figure 1.2. Figure 1.2 Fischer projections of glucopyranose and ribofuranos. Fischer projection of D-glucopyranose and D-ribofuranos in an B forms describing the open chain structure of compounds. If the hydroxyl group attached to a non-marked carbon possesses right-hand position, the compound will have a form while left-hand position of the hydroxyl group at a non-marked carbon gives rise to B form. In a Fischer projection, the topmost position is always occupied by the carbonyl group. D and L notation to the carbohydrates is given by comparing its Fischer projection structure to the glyceraldehyde structure. Glyceraldehyde is arbitrarily chosen as a standard for the D, L notation. Since glyceraldehyde contains an asymmetric carbon center, it exists as a pair of enantiomers. Figure 1.3. The structure of glyceraldehyde with its D form O on the right-hand side and L form O on the left-hand side are illustrated here. Hayward projection depicts the structural formula of monosaccharides as a cyclic structure having a simple three-dimensional perspective. The Hayward projection derived its name from the English chemist Sir Walter N. Hayward. The following characteristics best describe the Hayward projection. 1. Carbon is an embedded type of atom. There are a total of 6 carbon atoms in one circle and numbered 1 to 6. 2. The first carbon is called the anomeric carbon. 3. Hydrogen atoms attached to the carbon atoms are inherent. Figure 1.4 Hayward projections. Hayward's projection of monosaccharides having an B forms is shown above. The presence of the O group at the axial position gives A form, while at the equatorial position, the molecule is considered to have B form. Anomer. Anomer is a stereoisomer that mainly exists as a diastereomer. To be more appropriate, its cyclic structure possesses a different configuration only at the hemicidal having ketone group or hemiacetal having aldehyde group carbon. Hence, this carbon is named an anomeric carbon. The molecule is said to be A anomer if the structure is comparable to the one in which the hydroxyl group present on the anomeric carbon lies in the axial position of glucose. If the hydroxyl group occupies the equatorial position, then the molecule is known to possess AB anomer. Figure 1.5. Figure 1.3 Structural formulations of glyceraldehyde. Anomeric forms of fructofuranos are shown to have hydroxyl groups at the axial position A anomer and equatorial position B anomer. Another way to represent the cyclic structure of saccharides is the chair form and boat form. Figure 1.6. Saccharides in its chair conformation are said to have the lowest energy, whereas boat form is considered to have the highest energy. In chair conformation, the 6 hydrogens out of 12 are present in equatorial positions, and the other 6 hydrogens capture the axial position having their CH bonds in a parallel position concerning each other. The boat form carries 2 axial 1, 4 hydrogen atoms that generate steric strain. Axial position. In this position, there is an imaginary axis that is administered through the cyclic structure, and the sigma bonds lie at the axis parallel to it. Equatorial position. The sigma bonds are subjected to have a perpendicular position with respect to the imaginary axis mentioned above. Figure 1.5 Anomeric forms of fructofuranos. Figure 1.6 Chair and boat form representation of saccharides. Chair and boat form of sugar molecules is illustrated here. These forms are interconvertible by changing the axial and equatorial position of hydroxyl groups present in the molecule. Carbohydrate reactions. Carbohydrates being polyhydroxylated aldehyde ketone undergoes most of the reactions that are typical to molecules containing C equals O and O groups. Oxidation reactions. 1. Oxidation by bromine. Bromine water being the mild oxidizing agent selectively oxidizes the aldehyde group. It does not affect the hydroxyl or ketonic group present in the sugar molecule. With the help of this reaction, aldose and ketosaccharides can be distinguished as bromine water loses its reddish brown color in the case of aldose, whereas ketose cannot be further oxidized. The oxidation product of aldose is aldonic acid. Figure 1.7. Figure 1.7 Oxidation of glucose with bromine water. Glucose undergoes oxidation with bromine water with aldehyde being converted to the carboxylic acid group and thus shows itself as reducing sugar. 2. Oxidation by HNO3. Nitric acid is a strong oxidizing agent that oxidizes both aldehyde and alcoholic groups of aldose sugar. The product of the oxidation is known as alderic acid. Figure 1.8. Figure 1.8 Oxidation of glucose using nitric acid. Oxidation of glucose is shown in the presence of nitric acid and thus representing that glucose acts as to reduce sugar. 3. Oxidation reaction by Fellings. Benedict's and Tollens reagents. Sugars can be categorized as reducing and non-reducing ones based on their reactivity with these reagents. Saccharides that get oxidized by these reagents are said to be reducing sugar as the oxidant of the reagent produces ag+1 right pointing arrow ag0 and Cu+2 right pointing arrow Cu+1. This is advocated by the formation of a silver mirror or precipitation of cuprous oxide. These reagents, i.e., Fellings reagent, Tollens reagent, or Benedict's reagent, can be used to distinguish aldehydes from ketones. However, fructose can also reduce Tollens' reagents despite the absence of any aldehyde functionality. This can be explained on the basis that fructose has a tendency to isomerize under basic conditions, as provided in Tollens test, to produce a mixture of aldoses, glucose, and mannose, which ultimately reduces Tollens reagent. Figure 1.9. Figure 1.9. The reaction of monosaccharides with Tollens reagent. Monosaccharides having free aldehyde functionality reduce the silver ions present in Tollens reagent, and those having ketone moiety undergo an equilibrium to exist as aldose and hence confirm that monosaccharides are reducing sugars. Reduction reaction. Reduction of C equals O group of aldose and ketose sugar with not 4 under basic condition transformed into O groups of acyclic aldehdols. Figure 1.10. Figure 1.10. Reduction of monosaccharides. Monosaccharides also undergo reduction due to the presence of the C equals O group of aldose and ketose sugar to produce the alcohol functional group alditol. With the help of reducing agents such as NOD4, H2PT or PT or H2, ranini, D-glucose reduces to glucitol, commonly known as sorbitol. Sorbitol was first found in the barricades of mountain ash, Sorbus apicaria, and is about 50% as sweet as sucrose. It functions as a humectant and is also used in sugarless gums, toothpaste, hard candies, mints, and cough drops. It acts as preparatory material for the synthesis of vitamin C and sorbitan esters, emulsifiers as well. Figure 1.11. Reduction of glucose to glucitol or sorbitol. This figure represents the reduction of glucose to glucitol in the presence of a reducing agent. Fructose reduces to D-glucitol and D-mannitol under the pseudo reducing agent in the basic medium. Figure 1.12. Figure 1.12. Reduction of fructose. The reduction of fructose gives rise to the formation of glucitol and mannitol depending upon the arrangement of the O group generated from the reduction reaction. Y-lyososome formation. The reaction of saccharides with one equivalent of phenylhydrazine produces phenylhydrazone. Whereas in excess of phenylhydrazine, both C1 and C2 aldose, as well as ketose, react to form ososome of this hydrazone. Emil Fischer developed this reaction for the first time and identified aldose sugars those vary in a configuration only of the alpha carbon. The reaction of C2 ethanol with phenylhydrazone results in the formation of the same ososomes. Figure 1.13. Figure 1.13. Formation of ososome. The reaction of formation of ososome is given by both aldoses and ketoses due to the presence of an GT, C equals O functionality. Also, the C2 ethers of sugar molecules give rise to same ososome formation. Reactions of sugar due to hydroxyl groups. 1. Alkylation and alkylation of monosaccharides. The O group of monosaccharides reacts with acetic anhydride or acetyl chloride to form esters. Similarly, ether can be prepared using metal iodide, silver oxide. The nucleophilic character of the O group is rather poor. Hence, in the SN2 reaction, the use of silver oxide increases the leading tendency of the iodide ion. 2. Glycosides formation. Glycosides are cyclic acetal forms of sugars. The bond connecting the alkoxy oxygen and the anomeric carbon is known as the glycosidic bond. They are prepared by the acid catalyzed reaction of an alcohol with a pyranose or furanose. The alcoholic group in the reaction may be the O group of other monosaccharides. Both N-B glycoside are produced on reacting a single anomer with an alcohol moiety. The reason for the formation of both the products can be attributed to the formation a planar sp2 hybridized oxocarbon anion intermediate, which allows the attack of alcohol from both phases. Isomerization reaction. 1. Mutarotation. Pure AD-glucose and B-glucose vary from one another in their ability towards the rotation of the plane polarized light. AD-glucose rotates the plane polarized light by the specific rotation of +112, whereas its B anomer has a specific rotation of 18.7. However, they possess a tendency to quickly convert into a mixture of both the isomers at equilibrium and the aqueous solution. The specific rotation of one form decreases and the other increases, i.e., the specific rotation of a solution of AD plus glucose MP 146 degrees Celsius gradually declines from an initial value of +112.2 degrees to +52.7 degrees, while the BD plus glucose MP 150 degrees Celsius specific rotation gradually increases from an initial value of +18.7 degrees to +52.7 degrees. This change in specific rotation at equilibrium in the aqueous phase is called as mutarotation. The mutarotation can be observed in acid-base catalysis or neutral aqueous solutions at room temperature. Figure 1.14. Figure 1.14. Mutarotation of glucose. 0. 0. The mutarotation of glucose is shown in the figure through the conversion of A anomer to B anomer through either equilibrium mixture or open chain form. 2. Apomerization. When monosaccharides are heated in the basic aqueous solution, the change of stereochemical configuration at A carbon to C equals O group can be seen both in aldose and ketose form. The change can be observed as the removal of hydrogen from A carbon followed by the protonation of enolate ion. The reprotonation of the intermediate enolate ion on A carbon gives a mixture of the two epimeric monosaccharides with opposite stereochemical configurations at A carbon. The phenomenon of the formation of a mixture of epimeric monosaccharides is called apomerization. For example, in the presence of a base, D-glucose may be converted into D-mannose via the removal of hydrogen at C2 carbon followed by protonation of the enolate. Figure 1.15. Figure 1.15. Apomerization of glucose to mannose. The figure represents the apomerization of glucose to mannose involving the change in configuration of the O group through the formation of enolate ion. Monosaccharide derivatives. Monosaccharides act as an important precursor for the formation of a variety of biologically significant compounds or derivatives. The majority of monosaccharide derivatives can be categorized into three kinds, namely amino sugars, carboxylic acid sugars, and sugar alcohols. Amino sugars and N-acylated sugars. There is an amine functionality present in the amino sugars, unlike other sugars having hydroxyl as a functional group. Some sugars like derivatives of amino sugars such as sialic acid and N-acetylglucosamine, which do not contain the amino group formally, are also considered as amino sugars. Hence, N-acylated sugars, being derivatives of amino sugars, fall into the category of amino sugars. Glucosamine. Systematic name: 2-amino-2-deoxy-D-glucose. Molecular formula: C6H13 number 5. 2-amino-2-deoxy-D-glucohexose. C6H13 number 5 is an amino sugar biochemically synthesized from glucose by replacing its 2-hydroxyl group with the amino acid glutamine in the presence of an enzyme glucosamine synthetase. The amino sugar is a component of many structural polysaccharides and glycoconjugates, i.e., glycosylated lipids and proteins. Glucosamine also plays a role in the activation of the production of glycosaminoglycans, which can also be referred to as mucopolysaccharides, proteoglycans, and collagen. It also functions as a cushioning synovial fluid between the joints and in the formation of cartilage. N-acetylglucosamine, N-acetyl-D-glucosamine, or GLC-NAC, or NAG. Systematic name: 2-acylamino-2-deoxy-D-glucose. Molecular formula: C8H15 number 6. Chemically, it results from the reaction of acetic acid and glucosamine forming amide bonds and is substantial in numerous biological systems. N-acetylglucosamine comprises an essential component of a bacterial cell wall, which is made up of alternating units of GLC-NAC and N-acetylmoramic acid (MRNAC). The lactic acid residue of MRNAC provides the surface for cross-linking of these units with oligopeptides that result in a layered structure known as peptidoglycan. It serves quite a lot in the structural synthesis of polymer chitin and fungi cell wall, which governs the outer layers of crustaceans and insects. Hyaluronan is formed by the polymerization of glucuronic acid with GLC-NAC. GLC-NAC also imparts function in neurotransmission and nociceptive pain pathways. N-acetylglucosamine GLC-NAC functions in a variety of cells ranging from bacteria to humans, including mediation of cellular signaling. For example, in bacteria, GLC-NAC induces the components required for the colonization of human hosts. It leads to the formation of fibrins that imparts bonding to multi-drug exporter genes, host cells, and curly fibers that further stimulate biofilm formation. While, in mammals, GLC-NAC acts as a key sensor to identify the status of nutrients, which helps in cell cycle control, insulin signaling, and other essential processes. Galactosamine. Systematic name: 2-amino-2-deoxy-D-galactose. Molecular formula: C6H13 number 5. Galactosamine is also known as 2-amino-2-deoxy-D-galactose derived from galactose. This amino sugar acts as an important material for the synthesis of some glycoprotein hormones such as follicle-stimulating hormone (FSH) and luteinizing hormone (LH). Galactosamine can be found in pepper, C annuum green, orange, and yellow bell pepper, which makes it a potential biomarker for the consumption of these food products. It can also be marked as a unique E. coli metabolite. N-acetylgalactosamine (GalNAc). 2-acetylamino-2-deoxy-D-galactose. N-acetyl-D-galactosamine. Systematic name: 2-acetylamino-2-deoxy-D-galactose. Molecular formula: C8H15 number 6. It is a monoacetyl aminosaccharide derivative of galactose. In humans, the terminal carbohydrate comprises the antigen of blood group. It also forms an important constituent of brain heteropolysaccharides, making it essential for intercellular interaction and is concentrated in sensory nerve structures of both animals and humans. This saccharide can constrain the growth of some tumors. Norminic acid and sialic acid. Systematic name: norminic acid 5-amino-3, 5-deoxy-D-glycerol-D-galactonone 2-ulosonic acid. Molecular formula: C9H17N108. Norminic acid is considered a 9-carbon monosaccharide alcohol synthesized by the aldol condensation reaction of deaminosamine to amino-2-deoxyamminose and pyruvic acid. As a whole, the normal-substituted derivatives of norminic acid are known as sialic acids. The mammalian cell predominantly has N-acetyl-norminic acid. In general, the amino group in the saccharide contains either a glycol group or an acetyl group, whereas the hydroxyl substituents may be acetyl, methyl, lactyl, phosphate, and sulfate groups. They are the important components of oligosaccharide chains and polysaccharides such as mucins, glycoproteins, and glycolipids. N-acetyl-norminic acid, or NAC. Systematic name: R-2-acylamino-3O 1-carboxyethyl 2-deoxy-glucose molecular formula C11H19 number 8. This represents an ether moiety consisting of N-acetylglucosamine and lactic acid. It comprises an important component of the bacterial cell wall, which is made up of alternating units of N-acetylglucosamine GLC-NAC and N-acetyl-norminic acid (NAC) that are connected through one right pointing arrow forward glycosidic bonds. At the RNAC surface covered with lactic acid residue, these bonded entities get cross-linked with oligopeptides to produce a layered structure called peptidoglycan. Carboxylic acid sugars. Carboxylic acid sugars comprise two main categories, including aldonic acid sugars and uronic acid sugars. Aldonic acid sugars are produced by oxidation of carbonyl group present at the C1 position of monosaccharides, while uronic acid sugars can be resulted by C6 oxidation of monosaccharide sugars. Glucuronic acid. Systemic name: D-glucuronic acid molecular formula C6H10O7. Glucuronic acid is a well-known carboxylic acid sugar having a structure similar to that of glucose and possesses high solubility in water. Its C6 carbon possesses the functionality of the carboxylic group, and it is a major component of several glycoconjugates such as heparin, hyaluronic acid, and chondroitin sulfate. Glycolycerol lipids of glucuronic acid form the cell walls of bacteria. Being water-soluble, it often makes bonds, i.e. O-glycosidic bonds with different malicious substances present in different organs of an animal body, especially in the liver, to facilitate subsequent elimination, and with hormones to ease the transport. The process of making bonds is recognized as glucuronidation, and the compounds obtained are identified as glucuronides or glucuronicides. Gluconic acid. Systemic name: D-gluconic acid molecular formula C6H12O7. Gluconic acid is a carboxylic acid derivative of monosaccharide glucose, of which first carbon is oxidized to the carboxyl group. It forms gluconate ion C6H11O7. Minus on dissolving in water. The gluconic acid salts are also called gluconates. In an aqueous solution, some gluconic acid coexists in equilibrium with its cyclic ring ester form, i.e. gluconolactone. The major natural sources of gluconic acid include honey, fruit, wine, and kombucha tea, which are used as a food additive to regulate the acidity level. In an alkaline solution, it acts as a strong chelating agent. The gluconate ion gets involved in chelation with Ca2+, Al3+, Fe3+, and other metals, including lanthanides and actinides. The chelate complex of gluconate with zinc ion can be used to neuter male dogs. It also finds its use in the treatment of malaria on mixing with quinine. Sugar phosphates. Phosphorylated sugars comprise another significant kind of derivative of sugars. The hydroxyl groups of sugar molecules form an ester bond with phosphates. At the normal pH of a cell, generally, hydroxyl groups of the phosphate moieties remain in ionized form as O. Phosphorylated sugars play a key role as intermediates in the process of biosynthesis and energy generation. They act as building blocks for nucleotides and nucleic acids and are actively involved in the metabolism of sugars to release energy. Some important sugar phosphates are listed below. Glucose 6-phosphate. Molecular formula C6H13O9P. Glucose 6-phosphate is a glucose sugar having phosphorylation at the C6 position. It is also known as Robinson ester. This phosphorylated sugar is very commonly found in cells of the body as the vast majority of glucose gets phosphorylated inside the cells. It serves as a starting material for two main metabolic pathways, including the pentose phosphate pathway and glycolysis. It occurs in the form of starch or glycogen as stored energy or powerhouse of the cell. Generally, for most multicellular animals, it is present in muscles or liver as glycogen, while its glycogen granules are starch for most other organisms. Adenosine triphosphate ATP molecular formula C10H16N5O13P3. Adenosine 5' triphosphate ATP is also known as molecular currency. It acts multifunctional nucleotide having adenine substitution attached through an atom of the C1 carbon of ribose sugar. Saccharide, on the other hand, has a triphosphate substituent at the C5 position. ATP acts as a monomer for the synthesis of DNA and RNA. It initially gets converted to deoxyadenosine triphosphate (ADP) in the case of DNA. ATP is generated during the process of cellular respiration and photosynthesis. It is used up in a variety of cellular processes such as motility, biosynthetic reactions, and cell division. Enzymes present in the body also consume ATP for catalyzing a variety of biochemical reactions. For example, kinases utilize ATP for phosphorylation of lipids and proteins, while adenylate cyclase consumes it for the production of cyclic AMP as a second messenger molecule during signal induction pathways. It is transported in the form of chemical energy as an energy source to all the cells for various metabolic reactions. Cyclic adenosine monophosphate CAMP molecular formula C10H12N5O6P. Cyclic adenosine monophosphate 3'-5'. Cyclic adenosine monophosphate is also known as cyclic AMP or CAMP in short form and is derived from the ATP molecule in the presence of adenylyl cyclase, which is situated in the cell membranes. Various hormones, epinephrine and glucagon, help in the activation of the enzyme adenylyl cyclase. CAMP functions as one of the important molecules in various biological processes. It acts as a subsequent messenger for signal transduction within the cells, such as intracellular, used for intracellular signal transduction, such as conveying the consequences of hormones like adrenaline and glucagon, which cannot be obtained through the cell membrane. It also regulates the effects of glucagon and adrenaline, activates protein kinases, and controls the passage of Ca2+ through ion channels. Sugar alcohols. Sugar alcohol is known by different names such as a polyol, polyalcohol, or polyhydric alcohol. The sugar derived its name alcohol is a cardinal group, either aldehyde or ketone. Present in the reducing sugar gets reduced to primary if aldehyde is present originally, or secondary alcohol if ketone is present. The general formula of sugar alcohol is HHCHO N+1H. The most common sugars comprise lactitol, sorbitol, xylitol, mannitol, isomalt, and maltitol. Sugar alcohols are classified as generally recognized as safe, brought by the United States Food and Drug Administration (FDA). Their use in appropriate dosage does not cause any ailment like the decay of tooth or increase in blood sugar level, and hence can be used as food additives. Some important sugar alcohols are reviewed and demonstrated below. Ethylene glycol. Their use in appropriate dosage does not cause any ailment like the decay of tooth or increase in blood sugar level, and hence can be used as food additives. Some important sugar alcohols are reviewed and demonstrated below. Ethylene glycol. Systemic name: 1,2-ethane molecular formula C2H6O2. Ethylene glycol is a colorless, odorless syrupy liquid having a sweet taste. It is considered toxic and should not be ingested, which otherwise would lead to a medical emergency. It has many industrial and commercial applications. It acts both as a coolant and antifreeze, which means it protects the objects from freezing in cold temperatures while preventing from overheating in hot temperatures. The other important uses of ethylene glycol include the production of polyester fibers. Glycerol. Systemic name: 1,2,3-propanethiol molecular formula C3H8O3. Glycerol is a polyhydroxy compound commonly known as glycerin. Due to the tendency of hydrophilic hydroxyl groups to absorb water, it is considered hygroscopic in nature and possesses high solubility in water. It has low toxicity and finds use in pharmaceuticals due to its humectant and sweet-tasting properties. It is a major component of lipids. It acts as a precursor for the synthesis of phospholipids and triacylglycerols in adipose tissues in the liver. On consumption of stored energy by the body, it is released into the bloodstream and gets converted to glucose and hence providing energy for various cellular processes. Erythritol. Systemic name: 2R3S-butane-1,2,3,4-tetraene molecular formula C4H10O4. Erythritol is derived from erythrose. Erythritol occurs as a natural sugar alcohol and acts as a food additive of less sweet content, 60-65% of that of sucrose. It has much less caloric value than that of table sugar and hence does not affect blood sugar and tooth decay. Naturally, it can be found in fermented foods and fruits. Industrially, it can be obtained by fermentation of glucose in the presence of yeast, monoyl alkylines. It is associated with less risk of bloating in the stomach as it is partially digested by the intestinal bacterial, and rest gets excreted through urine and feces. Xylitol. Systemic name: xylopentane 1,2,3,4,5-pentyl molecular formula C5H12O5. Xylitol is a 5-carbon sugar alcohol that is derived from xylose. It is used as a substitute for sugar for many products and also in sugar-free products as well, such as mints, candies, and chewing gums, and hence is good for diabetic people. It is generally found in plant materials, including vegetables and fruits. It has sound medicinal properties and is used for patients with dental issues. It is very useful to prevent cavities in children of age 5 years and up. It helps in the prevention of the occurrence of ear infections commonly found in kids, though not effective to patients already having an ear infection. It also has the potential to treat osteoporosis. Mannitol. Systemic name: xylopentane 1,2,3,4,5-pentyl molecular formula C5H14O6. Mannitol is also known as manna sugar or mannatus. It is isolated from emissions of the flour ash, which is called manna due to its similarity with biblical food. It is a 6-carbon sugar and comprises one of the most produced carbon and energy storage compounds found in nature. It is produced by various organisms such as yeast, bacteria, fungi, lichens, algae, and many other plants. It is slightly absorbed by the body, and most of it is excreted out of the body through the kidney. Hence, it can be used to stimulate diuresis for excretion of toxic metabolites and prevention of acute renal failure before irreversible damage. It has many interesting medicinal properties. One of those makes it useful for handling brain mass and intracranial pressure when combined with hypertonic saline. Sorbitol. Systemic name: D-glucitol. Molecular formula: C6H14O6. Sorbitol, also known as glucitol, is obtained by the reduction of glucose 6-phosphate to sorbitol 6-phosphate by the catalytic reaction of the enzyme aldose 6-phosphate reductase. The reaction involves the conversion of an aldehyde group into a hydroxyl group, due to which it gets the name sugar alcohol. It is an isomer to sugar mannitol, and they have different orientations of hydroxyl group on the C2 position. It is often used in dietary foods such as ice cream, diet drinks, mints, sugar-free chewing gum, and cough syrups. Mostly fermentation of sorbitol cannot be done by bacteria but accepts Streptococcus mutans, which cause tooth decay. Sorbitol, though functions as a non-stimulant laxative but often leads to some abdominal pain, mild to severe diarrhea and gastric trouble if consumed in large amounts. Sorbitol is used as a humectant and thickener in modern cosmetics as well as in mouthwash and toothpaste. Due to its high refractive index, it can be used for making transparent gels for transparent formulations. Maltitol. Systemic name: 4-OAD-glucopyranosyl glucitol. Molecular formula: C12H24O11. Maltitol is a polyhydroxy sugar alcohol, a polyol, and can be used as a sugar substitute as it possesses less calories. It is used in confectionaries and making homemade sweets. As it is mildly absorbed by the intestine, it does not exert an inverse effect on tooth decay and blood glucose. However, it can be a causative agent of gastric distress if consumed in great quantities due to its water absorption from the intestine under the osmotic effect. It can be obtained by hydrogenation of maltose, which in turn can be produced from starch in the presence of enzymes on B amylases. Lactitol. Systemic name: 4-OBD-galactocopyranosyl D-glucitol. Molecular formula: C12H24O11. Lactitol is sugar alcohol composed of lactose and sorbitol. It retains 40% of the sugar sweetness and hence can be substituted for foods with low calorific value. Only a small portion of lactitol is absorbed in the small intestine in the form of glucose and sorbitol, whereas the rest passes to the large intestine, where it is fermented by colonic bacteria. Lactitol finds its applications in several local or food items. The higher stability of lactitol makes it suitable for use in bakery stuff and hence used in ice creams, sugar-free candies, chocolates, and cookies. It also functions as a prebiotic and hence stimulates good health of the colon. Lactitol possesses only 60% calories as compared with other carbohydrates. Disaccharides. Two monosaccharide sugar units are condensed together and linked through a glycosidic bond to give rise to disaccharides. These two units can be the same or different, resulting in homo or heterodisaccharide. Sucrose. Systematic name: D-glucopyranosyl 1 left right arrow 2 BD-fructofarnoside. Molecular formula: C12H22O11. Sucrose is also known as saccharose or common sugar and is a dimer of two monosaccharides, namely glucose and fructose. Its structure is formed through the linkage of the glycosidic bond of the C1 carbon of glucose to the C2 carbon of fructose monosaccharide. The glycosidic linkage in sucrose occurs between reducing end of both glucose and fructose, thus inhibiting further bonding to other saccharides. Hence, sucrose acts as a non-reducing sugar. Biochemically, it is synthesized by fructose 6-phosphate and uridine diphosphate glucose in the presence of enzyme catalyst sucrose 6-phosphate synthase. Lactose. Systemic name: 4-OBD-galactopyranosyl glucose. Molecular formula: C12H22O11. Lactose is most commonly known as milk sugar due to its notable presence in milk. In fact, it derives its name from milk in the Latin language along with ending up in nose is common to other sugars. Lactose is a disaccharide and is made up of B-glucose and BD-galactose fragments joined together through AB, one right pointing arrow 4 glycosidic linkage. In lactose, the lactose takes up B-pyranose form only, while glucose is free to take either A-pyranose or B-pyranose form. Hence, lactose can be named as either A-lactose or B-lactose, depending upon the anomeric form of the ring structure. Maltose. Systemic name: 4-OAD-glucopyranosyl glucose. Molecular formula: C12H22O11. Maltose is a disaccharide and is composed of two units of glucose joined together through an one right pointing arrow 4 linkage. However, the similar composition with B, one right pointing arrow 4 linkage results in the formation of cellulose, and with a one right pointing arrow 1 bonding between two glucose units yields trahulose, the different disaccharides. Maltose is also known as malt sugar or maltobiose. Further addition of glucose units results in the formation of maltotriose followed by the production of dextrins, having additional units of glucose, and eventually leads to starch synthesis. Hence, it can be categorized as reducing sugar. Cellobiose. Systemic name: AD-glucopyranosyl D-glucopyranoside. Molecular formula: C12H22O11. Cellobiose is a reducing disaccharide and is made up of two glucose molecules joined together through AB, one right pointing arrow 4 glycosidic bond. It can be hydrolyzed chemically by cation exchange resin or biochemically by bacteria to produce glucose. Cellobiose comprises H3 hydroxyl groups with 3 ether linkages in the structure and hence pertains to string and intermolecular hydrogen bonding among the molecules. Cellobiose functions as an indicator carbohydrate for malabsorption syndrome and Crohn's disease. Trahulose. Systemic name: AD-glucopyranosyl D-glucopyranoside. Molecular formula: C12H22O11. Trahulose is a disaccharide made up of two glucose units joined by one glucosidic bond. It is commonly known as stringulose or mycose. Various fungi, bacteria, plants, and invertebrate animals synthesize this disaccharide as a source of energy and to subsist water shortage and freezing conditions as it has very good water retention capability. It generally finds its use in cosmetics, the food industry, and pharmaceuticals. Industrially, it can be obtained from cornstarch. Trahulose is a non-reducing sugar as it exists in closed ring form, and aldehyde or ketonic form is not free to bind to other moieties. It has a large tendency to form hydrogen bonds, and hence, in an aqueous solution, it undergoes self-aggregation to form clusters of different sizes. Polysaccharides. Polysaccharides can be categorized as carbohydrates having multiple units of monosaccharide sugars joined together by glycosidic linkage. Most of the carbohydrates are found in nature as polysaccharides. The anomeric carbon atoms of all the polysaccharides get occupied in acetal formation. The absence of any free anomeric carbon atom in polysaccharides makes them non-reducing, and hence, they fail to respond to Tollens reagent. They also lack mutarotation. Polysaccharides can be classified based on the following criteria. 1. Based upon the monomeric units. 2. Based upon their biological role. Based upon their monomeric unit, they are classified as YY homoglycans, having the same monosaccharide as a monomeric unit are called homopolysaccharides or homoglycans. YY heteroglycans. The presence of more than one kind of monosaccharide results have to be known as heteropolysaccharides. Based upon their biological role, YY structural polysaccharides, cellulose. Chitin, arabinosilins, YY storage polysaccharides, glycogen, starch. Homoglycans. Homoglycans are also called homopolysaccharides. They are comprised of only one type of monosaccharide unit. Chitin, starch, cellulose, and glycogens are common examples of homoglycan and are made up of glucose sugar only. 1. Starch. Starch comprises one of the most abundant polysaccharides and is known as the prime storage carbohydrates of plants. It is a combination of polysaccharide amylose and amylopectin in the plant cell. The different components can be separated on the basis of their water solubility. Although starch is insoluble in cold water, it has a high tendency to bind water reversibly due to its hygroscopic nature. Different sources of starch contain different ratios and amounts of amylose and amylopectin, which in turn exert an effect on the properties of products made up of refined starch after processing. Most starch contain 80-90% amylopectin and 20-10% amylose. Both amylose and amylopectin produce D-glucose units upon acid. Amylose consists of a linear polymer of D-glucose in which a monomeric unit is linked with a 1-4 glycosidic linkage. Amylopectin can be categorized as a branched polysaccharide composed of amylose strands connected by 1,6 glycosidic bonds between the anomeric C1 of one amylose strand and C6 on another amylose strand. Enzyme A amylase present in saliva and small intestine can be able to break the 1-4 linkage of amylose and hydrolyzes it into maltose and maltotriose, whereas the 1-6 linkage of amylopectin can only be cleaved completely by the A-dextranase enzyme present in the small intestine in assistance with A amylase enzyme into NBD-glucopyranose. 2. Glycogen. Glycogen is a polysaccharide present in cells of humans and animals and it occurs in the stored form of sugar or powerhouse of sugar. It is structurally similar to amylopectin except that it is highly branched, where branches occur at every 8 to 12 monosaccharide units. It is known for its highly branched structure providing many glucose and group residue sites, which on hydrolysis gives glucose units immediately needed for metabolism. The highly branched structure of glycogen provides more active sites for the enzyme attack and is hence easily available as a source of glucose in the case of need. Branched structures, due to the presence of many glucose end groups, rarely provide several attacking sites for the enzyme as compared to the linear polymeric glucose structure. The major site for the storage of glycogen is the liver and muscle. The concentration of glycogen in the liver is higher than in the muscles. Glycogen can be produced using monomers of UDP glucose in the presence of enzyme glycogen synthase, and then a chain of glycogen lengthens progressively. Both glycogen and protein and glycogen synthase are needed for the activation of glucose as UDP glucose. The protein glycogen initiates the formation of glycogen while glycogen synthase lengthens that existing chain as the latter cannot start the synthesis of glycogen but can only increase the chain length. UDP glucose, in turn, can be synthesized using glucose 6-phosphate in two steps. The first step includes the conversion of glucose 6-phosphate to glucose 1-phosphate in the presence of the enzyme phosphoglucomatase. The second step comprises the subsequent conversion to UDP glucose with the release of pyruvate in the presence of the enzyme uridine triphosphate UTP. 3. Cellulose. Cellulose classified structural homoglycan made up of linearly joined D-glucose unit by 1,4 glycosidic bond. Most abundant organic polymer provides structural integrity and strength to the primary cell wall of green plants and many forms of algae and mycetes. Some species of bacteria secrete it to produce biofilms. Humans and other mammals do not have the enzyme B-glucosidase needed to hydrolyze cellulose. So, this class cannot obtain glucose as energy serving part directly from cellulose. It is an insoluble molecule in an aqueous solution and consists of a linear polymer of B1 to 4 D-glucopyranose units approximately in the range of 2,000–14,000 residues. The multiple hydroxyl groups of either the same unit or different units of fully equatorially arranged B-linked glucopyranose residues provide high tensile strength and rigidity to the structure and hence stabilize the chair conformation. 4. Chitin and chitosan. Chitin comprises the second most abundant natural polysaccharide after cellulose on Earth and is composed of B1 to 4 linked to acetyl 2-deoxy-B-D-glucose and acetylglucosamine. It is structurally identical to cellulose but has a semi-groups NHCOCH3 at the C2 positions. Chitin, which occurs in nature as an aminopolysaccharide polymer, is a major building material that gives strength to the exoskeletons of crustaceans, insects, and the cell walls of fungi. Chitin exists in the form of two allomorphs, namely the and B forms, that can be characterized by infrared and solid state NMR spectroscopy, together with X-ray diffraction. The third allomorph of chitin has also been described as G-chitin, but detailed studies show that it is just a variant of A allomorph. Chitosan is a linear polymer of A1 to 4 linked to amino 2-deoxy-B-D-glucopyranose, and N-decylated chitosan is a copolymer of N-acetylglucosamine and glucosamine. The degree of decylation is a measure of the extent of decylation. The presence of amino groups at C2 positions of chitosan makes it more versatile compared to the chitin structure. Heteroalkanes. Heteroalkanes are also known as a heteropolysaccharide and are made up of two or more different monosaccharide units. Chemically, they are formed mostly from repeating disaccharide units. The most common heteroalkanes include hyaluronic acid, chondroitin sulfates, and dermatan sulfates, which are usually present in the extracellular matrix. The repeated units of disaccharide acid, sugar, and amino sugar result in the formation of these heteropolysaccharides. Hence, they have been named glucosaminoglycans, GAGs, or mucopolysaccharides. Chondroitin sulfate. Chondroitin sulfate is a type of sulfated glycosaminoglycan, GAG, built up of a chain of alternating sugars called N-acetyl-galactosamine and glucuronic acid. A chondroitin chain can contain over 100 sugars, each of which can be sulfated in different places and amounts. Chondroitin sulfate is a substance found in the cartilage that surrounds the body's joints. Chondroitin sulfate chains are unbranched polysaccharides with two monosaccharides that alternate, D-glucuronic acid, GLCA, and N-acetyl-D-galactosamine, GalNAc. Some GLCA residues are epimerized to form hyaluronic acid, EDOA, which is then referred to as dermatan sulfate. Dermatan sulfate. Dermatan sulfate was formerly known as mucopolysaccharide. It's a type of glycosaminoglycan found mostly in the skin, but also in heart valves, blood arteries, the lungs, and tendons. Coagulation, cardiovascular disease, carcinogenesis, infection, wound repair, and fibrosis may all be affected by dermatan sulfate. In various mucopolysaccharidosis conditions, dermatan sulfate accumulates improperly. Choroidin sulfate. Choroidin sulfate is a type of sulfated glycosaminoglycan found in the cornea, cartilage, and bone, among other places. It's a big, highly hydrated structural polysaccharides molecule that acts as cushioning in joints. There are two major categories. The cornea contains type 1, while cartilage has type 2. Heparin. Heparin is a member of the glycosaminoglycan family of carbohydrates, which includes the closely related molecule heparin sulfate. It is widely used as an injectable anticoagulant and has the highest negative charge density of any known biological molecule. It can also be used to form an inner anticoagulant surface on various experimental and medical devices such as test tubes and renal dialysis machines. Heparin sulfate. Heparin sulfate HS is a linear polysaccharide found in all animal tissues. It occurs as a proteoglycan PG in which 2 or 3 HS chains are attached in close proximity to the cell surface of extracellular matrix proteins. It is in this form that HS binds to a variety of protein ligands and regulates a wide variety of biological activities, including developmental processes, angiogenesis, blood coagulation, and tumor metastasis. Hyaluronan. Hyaluronan, also called hyaluronic acid or hyaluronate, is a non-sulfated glycosaminoglycan distributed widely throughout connective, epithelial, and neural tissues. Hyaluronan is a polymer of disaccharides composed of D-glucuronic acid and D-N-acetylglucosamine, linked together via alternating B1, 4 and B1, 3 glycosidic bonds. Hyaluronan can be 25,000 disaccharide repeats in length. Polymers of hyaluronan can range in size from 5,000 to 20 million Daltons in vivo. It is one of the chief components of the extracellular matrix, contributes significantly to cell proliferation and migration, and may also be involved in the progression of some malignant tumors. Glycoconjugates. Glycoconjugates comprise a very important class of compounds in biological science and consist of covalently linked saccharides with different chemical species such as proteins, peptides, lipids, and saccharides. The carbohydrate linked with protein is further classified into two categories depending upon their carbohydrate content. Proteoglycan with high carbohydrate content as well as oligosaccharides linked with small polypeptide. Deep glycoproteins occur in diverse structural form with great potential to carry out a variety of functions and have many functions. They remain bound to membranes and well as may exist in extracellular fluids in its soluble form. The saccharide attached covalently to different moieties such as protein lipids plays a fundamental role in the function of glycoconjugates. The example of the integral role of saccharides can be accomplished by glycoprotein NCAM involved in different roles such as cell adhesion, synaptic plasticity, neurite outgrowth, memory and learning abilities. The structural detail of the saccharides linked to the protein and the glycoconjugates determines the cell binding. Proteoglycans can be categorized depending upon the relative size of saccharides and the nature of glycosaminoglycans and can be summarized as below table 1.2. Small proteoglycans, large proteoglycans, glycosaminoglycans. Vicunin, 25 kDa neurocan, 136 kDa abracan, 220 kDa, the major. Proteoglycan in cartilage. Brevican, 145 kDa. Chondroitin sulfate. Decorin, 36 kDa beta-glycan, 38 kDa ursican, 260 to 370 kDa, present in many adult tissues such as skin and blood vessels. Chondroitin sulfate, dermatan sulfate. Fibromodulin, 42 kDa. Lumican, 38 kDa choroidin sulfate. Testican, 44 kDa prolectin, 400 to 470 kDa heparin, chondroitin sulfate table 1.2 different types of proteoglycans. Types of glycoproteins. As mentioned above, glycoproteins can be broadly categorized into two kinds on the basis of their position of glycosylation as N-glycosylation and O-glycosylation. 1. N-glycosylation. It involves the addition of sugar moieties glycans to the amide nitrogen of the residual chain of asparagine ASN. This linkage occurs usually in eukaryotic proteins and is important to both their structure and function. It also functions in archaea but is rarely noticed in bacteria. The type of N-glycosylation can be varied across different species, and their nature can be described by the protein and their expression in the cell. 2. O-glycosylation. In the case of attachment of monosaccharide N-acetyl-galactosamine, it is transferred from root GalNAc uridine 5' to acetyl-galactosamine to the polypeptide chain to the hydroxyl of S or THR residue of fully folded protein, and the process is catalyzed by the enzyme GalNAc transferase. Elongation and modification of O-GalNAc can be done by the attachment of other carbohydrates such as sialic acid and galactose and different compounds containing methyl, acetyl and other chemical groups. 3. O-fucose. Unlike the O-gainic, the addition of fucose to serine or threonine residue takes place in the endoplasmic reticulum and the process of addition is catalyzed by the enzyme fucosyltransferases. After the addition of O-fucose, it is modified and elongated by the addition of other monosaccharides, namely GLC-NAC, galactose and sialic acid. In the case of Notch protein, O-fucosylation occurs in between the second and third conserved cysteines. The protein has epidermal growth factor (EGF) domains which are O-fucosylated. Sequential addition of galactose, N-acetylglucosamine, and sialic acids to O-fucose elongates the chain to produce tetrasaccharides in the instance of EGF-like repeats. On the other hand, in the case of thrombus-bonded repeats, the addition of glucose produces disaccharides only. Both of these fucosyltransferases generally function in the Golgi apparatus but are located in the endoplasmic reticulum. 4. O-glucose. O-glucosylation modification, same as O-fucosylation, takes place in the endoplasmic reticulum catalyzed by the enzyme O-glucosyltransferases. In the Notch protein, the enzyme O-glucosyltransferases adds to the serine residue between the first and second conserved cysteines of X-like repeats. O-glycosylation is important for proper folding of the EGF domain in Notch protein. 5. O-N-acetylglucosamine, OGLC-NAC. The addition of OGLC-NAC to serines or threonines takes place in the presence of enzyme OGLC-NAC transferase. The process differs from O-glycosylation as in this modification the sugar moiety is attached and removed several times on the core protein with the help of two enzymes namely OGLC-NAC transferase OGT for the addition and O-glucanase OGA for the removal respectively. Usually, threonine serine kinesis phosphorylate OGLC-NAC, but it actually takes place on occurring on threonine and serine residues. The process of attachment of OGLC-NAC occur in altered with phosphorylation, which means if O-glycosylation occurs, phosphorylation does not and vice versa. 6. GPI anchor. GPI anchor comprises a unique form of glycosylation process. It serves to link the protein through a glycan chain to the hydrophobic lipid anchor. 7. O-mannose. The dolichopene mannose acts as a donor molecule and facilitates the addition of mannose to the serine or threonine residue of protein. The process starts in the endoplasmic reticulum and further addition of sugar gets accomplished in Golgi apparatus. Further modification of O-mannose can be done by the addition of different monosaccharides such as ribitol, xylose, and glucuronic acid. Though the process appears to be restricted to the fungi previously, but later it occurs in eukaryotes as well as in archaea. A-dystroglycan is an example of an home-insulated protein found in humans. The sugar code. Lectin. Lectins are sugar-binding proteins that possess an affinity to bind with specific sugars only. They can also bind to soluble sugar molecules of glycolipids or glycoproteins. Lectins are usually non-enzymatic in nature. Lectins generally have a plant origin and can be found abundantly in grains and beans. Lectins are majorly comprised of that specific portion of the seed that grows into leaves on sprouting of the plant. Lectins can be classified in various ways depending upon a different basis. On the structural basis, there are 4 major categories of lectins such as 1. Concanavalin C. 2. Erythrin C. 3. C-type N. 4. Ulixeuropeous sugar binding ability to certain sugars comprises another classification basis, and those sugars are 1. Sialic acids. 2. Glucose or mannose. 3. L-fucosen. 4. N-acetyl-D-galactosamine and galactose. Lectin is known to have a characteristic role in the innate immune system. Lectin binding with mannose saccharide helps in defending the invasion of a microorganism by recognizing the carbohydrate component found on pathogens. Their role in animals also includes cell adhesion and regulation of blood protein level due to their binding with soluble extracellular and intercellular glycoproteins. Lectins are known for its recognition ability at both cellular and molecular platform and hence are associated with different biological recognition processes in the body, such as in proteins, carbohydrates, and cells. One such example includes the mannose 6-phosphate receptor that senses hydrolytic enzymes having lectin residue and subsequently regulates the delivery of these proteins to the lysosomes. They can also be used to recognize the changes in cell surface structural integrity categorized into B and T lymphocytes, and hence any viral or parasitic infection can be diagnosed using lectins. Lectins also find a specific role in the analysis of blood groups. They contribute significantly as the carriers for the delivery of pharmaceutical drugs in the body due to their ability to recognize cell surface receptors. Lectins possess significant agglutinating and adhesive properties and help microorganisms to adhere to host surfaces, and also play a significant role in pathogenic and symbiotic interactions among them. The major advantages of lectins include its stability and ability to perform functions when present in even small concentrations. Due to their numerous properties, specifically recognition ability, they play a vital role in microbiology. They act as naturally occurring histochemical and cytochemical unique tools and hence possess potential significance for its use as diagnostic reagents in clinical microbiology. The various other applications of lectins comprise its use in inflammation, serology, tumor marker, and food and agriculture. Glycome. Glycome represents the entire carbohydrate content present in the cells of the human body, either in the free state or in complex form. It comprises the most complex integrity of nature. The detailed study of all glycan structures present in the cells or organism constitutes the branch of study called glycomics, which in turn comprises a part of glycobiology. Similarly, proteomes comprise the study of protein structures in the body. 4. N-acetyl-D-galactosamine and galactose. Lectin is known to have a characteristic role in the innate immune system. Lectin binding with mannose helps in defending the invasion of microorganisms by recognizing the carbohydrate component found on pathogens. Their role in animals also includes cell adhesion and regulation of blood protein level due to their binding with soluble extracellular and intercellular glycoproteins. Lectins are known for its recognition ability at both cellular and molecular platform and hence are associated with different biological recognition processes in the body, such as in proteins, carbohydrates, and cells. One such example includes the mannose 6-phosphate receptor that senses hydrolytic enzymes having lectin residue and subsequently regulates the delivery of these proteins to the lysosomes. They can also be used to recognize the changes in cell surface structural integrity categorized into B and T lymphocytes, and hence any viral or parasitic infection can be diagnosed using lectins. Lectins also find a specific role in the analysis of blood groups. They contribute significantly as the carriers for the delivery of pharmaceutical drugs in the body due to their ability to recognize cell surface receptors. Lectins possess significant agglutinating and adhesive properties and help microorganisms to adhere to host surfaces, and also play a significant role in pathogenic and symbiotic interactions among them. The major advantages of lectins include its stability and ability to perform functions when present in even small concentrations. Due to their numerous properties, specifically recognition ability, they play a vital role in microbiology. They act as naturally occurring histochemical and cytochemical unique tools and hence possess potential significance for its use as diagnostic reagents in clinical microbiology. The various other applications of lectins comprise its use in inflammation, serology, tumor marker, and food and agriculture. Glycome. Glycome represents the entire carbohydrate content present in the cells of the human body, either in the free state or in complex form. It comprises the most complex integrity of nature. The detailed study of all glycan structures present in the cells or organisms constitutes the branch of study called glycomics, which in turn comprises a part of glycobiology. Similarly, proteomes comprise the study of protein structures in the body. The glycomes are even more complex and diverse than proteomes, and it further gets complicated by the interaction of carbohydrates among themselves and with proteins in the organisms. In the human body, the size and magnitude of glycomes present exceed the number of proteomes coded by the genomes. That makes the scientists research in the area of carbohydrates due to the well-known fact that sugars can be found everywhere as all the cells in the body are coded by sugars. They are indulged in the pathophysiology of all major diseases. They have considerable potential in the discovery of biomarkers, especially in the treatment of cancer. In various diseases like cancer, inflammation, and diabetes, the cells have been found to contain unusual glycosylation patterns. Also, the changes in structural properties of glycans are not only associated with the disease state but with body index mass, aging, smoking, and other factors as well. Mapping and characterizing the glycomes, the body sugar comprise an emerging area of interest of scientists at the international level, and in this regard, the Human Glycome Project is one of the initiatives. In this project, scientists are aiming to unfold the complex structural and functional integrity of glycoconjugates and hence the multicellular system. Glycosylation of proteins comprises one of the most multifaceted post-translational reformations of proteins. Any changes in glycosylation, especially for site-specific glycosylation, may lead to unusual outcomes analogous to mutations caused due to altered coding of genes. Hence, a systematic study of glycomes would open up multiple new opportunities to understand human health and to control life for betterment. Structure and functions of protein amino acid family. Proteins are involved in virtually every cellular process, exhibiting an endless diversity of functions. Proteins are the most abundant biological macromolecules, occurring virtually in every cell. Proteins also occur in great variety. Thousands of different kinds may be found in a single cell. Proteins are made of amino acids, wherein each amino acid is joined to the neighboring amino acid by a peptide bond. Proteins are composed of 20 standard amino acids. Asparagine was the first amino acid to be discovered in 1806. The most recent is threonine, identified in 1938. All 20 of the common amino acids are A amino acids, meaning that the A carbon is attached to a carboxyl group and an amino group. Figure 1.16. The R group of each amino acid has a different structure, size, and electric charge, which in turn influences the solubility of the amino acids in water. Figure 1.16 a general representation of amino acid. The chiral center is the A carbon. It is attached to an amino group and a carboxyl group. The R group varies for each amino acid. The A carbon of any amino acid is attached to an amino group, a carboxyl group, an R group, and a hydrogen atom, making the A carbon atom the chiral center. The orbitals of these groups are in a tetrahedral arrangement around the A carbon atom. The 4 different groups can occupy 2 unique spatial arrangements, resulting in each amino acid giving rise to 2 possible stereoisomers which are non-superimposable. Apart from these, there are many less common amino acids. Figure 1.17 representative images of stereoisomers in A amino acids. 1. The stereoisomers of the simple amino acid alanine, LND-alanine, are non-superimposable mirror images of each other, also called enantiomers. B, C, shown here are 2 different conventions that are commonly followed for showing the configurations in the space of stereoisomers. 2. To specify the configuration of amino acids, a special nomenclature has been developed, known as the D-L system. Glyceraldehyde is taken as a template model. If the carboxyl group of alanine occupies the same position around the chiral carbon as that of the aldehyde group of L-glyceraldehyde, the configuration is denoted as L-alanine. The same is the case for D amino acids. However, only L amino acids are found in physiological proteins. Rarely some D amino acid residues have been found in some small bacterial cell wall peptides and certain peptide antibiotics. Classification of amino acids. Standard amino acids. The standard amino acids can be classified based on the polarity of the side chains R groups. They are classified into 3 groups, namely amino acids having nonpolar side chains, amino acids having uncharged polar side chains, and amino acids having charged polar side chains. 1. Amino acids having nonpolar side chains. Some amino acids contain nonpolar side chains R groups such as H and glycine, CH3 and alanine, CHCH3 2 and valine, CH2CH3 2 and leucine, CH3 CH2CH3 and isoleucine, etc. There are 9 standard amino acids with nonpolar side chains, namely glycine, alanine, valine, leucine, isoleucine, proline, methionine, phenylalanine, and tryptophan. These amino acids are mostly found in the interior of the proteins, which function in an aqueous environment and on the surface of such proteins, which interact with lipids. 2. Amino acids having uncharged polar side chains. Some amino acids contain uncharged polar side chains, R groups, such as O groups present in the side chains of serine, threonine and tyrosine, SH group in the side chain of cysteine and CONH2 groups in the side chains of asparagine and glutamine. These amino acids are mostly found on the outside of proteins that function in an aqueous environment and in the interior of membrane-associated proteins. 3. Amino acids having charged polar side chains. Some amino acids contain charged polar side chains, R groups, such as acidic or negatively charged amino acids and basic or positively charged amino acids. Aspartic acid or aspartate and glutamic acid or glutamate, being negatively charged amino acids, have acidic side chains at physiological pH 7.4. Lysine, arginine and histidine, being positively charged amino acids, have basic side chains at physiological pH 7.4. These amino acids are mostly found on the outside of proteins that function in an aqueous environment and in the interior of membrane-associated proteins. Non-standard amino acids. All amino acids which are not incorporated ribosomically into the proteins are known as non-standard amino acids. There are certain non-standard amino acids which are produced after post-translational modification of the standard amino acids. These modifications are specific for those proteins to function properly. For example, 4-hydroxyproline and 5-hydroxylisine are formed as a result of modifications in proline and lysine, respectively. 1. 4-hydroxyproline is a derivative of proline, and 5-hydroxylisine is a derivative of lysine. Both are found in collagen, a fibrous protein of connective tissues. Additionally, 4-hydroxyproline is also found in some plant cell wall proteins. 2. 6-methylysine is a constituent of myosin, which is a contractile protein. 3. G-carboxyglutamate is found in prothrombin, a blood clotting protein and other CA2+ binding proteins. 4. Desmosine is an unusual derivative of 4-lysine residues. It is commonly found in the connective tissue in the fibrous protein known as elastin. 5. Selenicystine is introduced during protein synthesis. It is involved in the catalytic activity of enzymes like glutathione peroxidase and 4-ate dehydrogenase. 6. Apart from the rare amino acids listed above, there are some amino acids that are modified only transiently to change or regulate the functionality of a protein. For example, 6. Apart from the rare amino acids listed above, there are some amino acids that are modified only transiently to change or regulate the functionality of a protein. For example, the addition of methyl, phosphoryl, acetyl, ATP ribosol, or other groups. Ionization pattern of amino acids. When an amino acid with a neutral R group is dissolved in water pH 7.0, it exists as a dipolar ion which is also known as deuterium German 4 hybrid ion, meaning that it can act as either an acid or a base. Figure 1.18. This dual acid-base nature is referred to as amphoteric, and such substances are called ampholytes. Figure 1.18. Ionization of amino acid. Diagram showing the ionization pattern of amino acid with a non-ionizable R group. It is important to note the amphoteric behavior of amino acids. Since we now know that amino acids ionize, the characteristic pH of an amino acid at which the net electric charge is 0 is known as the isoelectric point or isoelectric pH, designated as pi. For glycine, which has a non-ionizable group H in its side chain, the isoelectric point is calculated by taking into account the mean of the two pKa values. The pi can also give us information about the migration of an amino acid in a particular electric field. The amino acid at a pH above its pi will have a net negative charge. Similarly, it will have a net positive charge at pH below its pi. However, amino acids with ionizable R groups have different titration curves for the different ionization stages. The pi hence depends on the nature of the R group. Table 1.3. Amino acid abbreviated. Names. Single letter code. pKa1. COOH. pKa2 NH3+. pKa3. R group pi. Names. Single letter code. pKa1. COOH. pKa2 NH3+. pKa3. R group pi. Lysine K 2.188.95 10.539.74 arginine R 2.179.04 12.48 10.76 histidine is H 1.829.176.007.59. Table 1.3 classifications of amino acids. In the case of proteins or peptides, there is only one free amino group and one free carboxyl group, which ionize normally as in free amino acids, even though the ionization constants are dramatically different. These peptides, like amino acids, also have their own characteristic titration curves and isoelectric pH pi at which they do not migrate in an electric field. These peptides, like amino acids, also have their own characteristic titration curves and isoelectric pH pi at which they do not migrate in an electric field. Protein structure. Two peptide bonds between three amino acids form a tripeptide. Four amino acids have three peptide bonds between them and form a tetrapeptide, and so on. When a few amino acids are joined, they are called oligopeptides. When several amino acids come together, we call it a polypeptide. There are some multi-unit proteins which have two or more polypeptides that are associated non-covalently. If the different polypeptides are identical, it is an oligomeric protein, and the identical units are referred to as protomers. Hemoglobin, for example, has four polypeptide subunits, two identical chains and two identical chains, all the four of which are held together via non-covalent interactions. Why are there so many different proteins with so many diverse functions? The basis of this underlies the structure. The structure of a protein defines its functionality. Protein structure is also generally referred to in terms of conformation. Conformation is the shape of a protein in space without disrupting any covalent bonds. The most stable conformation, which has the lowest Gibbs free energy G, which is functionally active, is referred to as native conformation. What factors influence the most stable conformations of a typical protein? The structure-function relationship of proteins can be summarized as follows. 1. Protein structures are determined by its amino acid sequence. 2. Function of a typical protein depends on its structure. 3. Most isolated proteins exist in one or a small number of structural forms that are stable. 4. Non-covalent interactions stabilize the specific structures of a protein. 5. Some common structural patterns help to understand protein architecture. 6. Protein structures undergo changes in conformation, which may or may not affect their function. Protein structure can be divided into 4 degrees of complexity. 1. Primary structure. This is the linear sequence of the amino acids in a polypeptide. The amino acids in a polypeptide chain are bound by two types of covalent bonds, peptide bonds and disulfide linkages. The primary structure of insulin is shown below as an example, figure 1.19. Mostly, the sequence of the protein dictates its functions, although some regions exist, which even if changes are removed, will not significantly affect the function of the protein. More than 3,000 different proteins are produced by Escherichia coli. A human has approximately 25,000 genes encoding a much larger number of proteins. In both of them, each type of protein has a specific amino acid sequence which gives it a specific unique three-dimensional structure. This structure is responsible for its function. The sequence of the first polypeptide to be discovered was that of insulin by Frederick Sanger. Nowadays, the amino acid sequences of proteins can be derived from the genome databases indirectly from the DNA sequences. Figure 1.19 Primary structure of insulin. Insulin is a simple peptide hormone consisting of 51 amino acids. Note the presence of connecting disulfide linkages. 2. Secondary structure refers to the stable arrangements of amino acid residues giving rise to recurrent structural patterns. The next thing after knowing the sequence of a protein is to study its structure. The protein backbone has numerous bonds around which free rotation is possible. The secondary structure deals with the arrangement of the peptide bonds in a protein. Different types of secondary structures are A helix. This is the most abundant secondary structure. Up to approximately 35% of residues in a protein are in an A helix arrangement. Figure 1.20. This helix has 3.6 residues per turn and a pitch of 5.4A. The carboxyl group of ITH amino acid forms hydrogen bonding with the amino group of I plus 4TH amino acid residue. They exhibit interstrand H bonding wherein C equals O groups are aligned parallel to the peptide axis. Side chains, on the other hand, are pointing away from the axis. It is to be noted that all the amino and the carboxyl are hydrogen bonded, except the first ones. Hence, the ends of the helix are polar, making the A helix amphipathic. 310 helix and pi minus helix are some variations of A helix, which are either very tightly or loosely packed, respectively. The amino acids preferentially located in the core of the helix are ALA, Lu, Met, Glu, Arg, and Li. On the other hand, there is Pro since its side chain is linked to AN and it has no amino group to hydrogen bond, and it also has a rigid structure due to its amino ring light since it does not have a side chain TR and SR. A common secondary structure in proteins. Alpha helix is mainly found in transmembrane proteins due to its amphipathic nature. B sheet. The basic unit is AB strand. It can either be parallel or antiparallel, depending on the direction of the interacting B strands. Figure 1.21. Unlike the A helix, hydrogen bonding in AB sheet is interstrand, i.e. the side chains from the adjacent residues are present on the opposite sides of the sheet. Hence, they cannot interact with one another. Parallel B minus sheets are structures wherein the adjacent amino acids are placed in the same direction, and antiparallel B minus sheets are sheets in which the amino acids in consecutive strands are alternating in their directions. B-sheet structures are mostly formed by amino acids with bulky side chains like TIR, TRP, EL, VAL, THR, CYs and P, whereas amino acids such as ASP, ASN, and S are compete with H bonds of B turns loops and are very poor helix formers. Figure 1.21 parallel versus anti-parallel beta sheets. Figure 1.20 alpha helix. Schematic representation of B sheet wherein the strands are aligned parallel and anti-parallel. Anti-parallel beta sheets are more stabilized. 3. B turns. Apart from the A helix and the B sheet, there are other secondary structures found in proteins known as turns or loops. They are basically structures wherein the polypeptide chain can reverse its direction. These structures serve to connect the successive sheets or helices to one another. B turn is mostly responsible for linking the adjacent ends of an anti-parallel B sheet. It has 4 amino acid residues, with the cardinal oxygen of the first amino acid residue hydrogen bonding with the amino group hydrogen of the fourth amino acid residue. The remaining 2 amino acids have no role in any inter-residue hydrogen bonding. Several types of turns are found, each defined by the bond angles linking the 4 amino acid residues that make up the particular turn. Glide. Glide, being small and flexible in structure and process, it can easily take the cis configuration, helps in maintaining a tight turn. Beta turns are often found near the surface of a protein, where the peptide groups of the central 2 amino acid residues in turn can hydrogen bond with water. Considerably less common is the turn, a 3 residue turn with a hydrogen bond between the first and third residues. Protein domains. These are basically specific arrangements of 2 or more secondary structure elements usually responsible for a specific function, like metal ion binding or DNA binding. They can be part of a larger functional and/or structural assembly known as a domain. 1. Helix turn helix HTH. This is a structural motif responsible for DNA binding. In this type of secondary structure, 2 A helices are joined by a loop. This is also known as a helix loop helix HLH motif. One helix is responsible for recognizing the nucleotide sequence by inserting it into the DNA groove, and the other helix functions to stabilize the configuration. 2. EF hand. This particular structure is a common feature of many calcium binding proteins like calmodulin. 3. B hairpin. This structure is composed of 2 antiparallel B strands joined together by loop. 4. Greek key motif. This motif consists of 4 adjacent antiparallel B strands. 5. B alpha B fold. This structure has parallel B strands connected by an A helix. This finds in place in proteins with parallel B strands, e.g., triphosphate isomerase. 6. Rossmann fold. This is a specific example of AB minus a minus B fold wherein 3 parallel B sheets are connected via 2 helices. This is found in nucleotide binding protein domains. Tertiary structure involves the 3-dimensional folding of a polypeptide. It comprises of protein domains, which are structures that can fold independently into a stable tertiary structure and are the fundamental units of the tertiary structure of a polypeptide chain. For each the catalytic domain of protein kinase, the binding pocket of a ligand. The arrangement of the different polypeptide subunits of a protein in space is referred to as quaternary structure. The difference in the free energy between the folded and unfolded states of proteins in physiological conditions is normally found to be in the range of 20 to 65 kilojoules per mole. A polypeptide chain can take up various conformations, and hence the unfolded protein has high conformational entropy. This entropy and the hydrogen bonding interactions with water helps to maintain the unfolded state. Disulfide linkages also help in stabilizing the protein structure. These are quite common in eukaryotes, but relatively rare in bacterial proteins. However, thermophilic bacteria, as well as the archaea, typically have disulfide bonds containing proteins, which stabilize them. This could be a response or an adaptation to life at high temperatures. For all proteins of all organisms, weak interactions are mainly responsible for the folding of polypeptide chains into their secondary and further tertiary structures. Multiple polypeptides are associated to form quaternary structures, and these weak interactions are involved in this process. 200 to 460 kilojoules per mole of energy is required to break a single covalent bond. Weak interactions, on the other hand, require 0.4 to 30 kilojoules per mole. Even though covalent bonds are stronger than weak interactions, because the weak interactions outnumber the covalent bonds, these interactions, mainly hydrophobic interactions, act as a stabilizing force that is required to maintain the protein structure. The increased order of the water molecules in the solvation layer indicates entropy of the water is less. However, when nonpolar groups come together, because each group is no longer fully exposed to the solution, the extent of the solvation layer decreases, leading to a favorable increase in entropy. This is the reason why hydrophobic amino acids are mostly present in the interior of a protein. The amino acid sequences of most proteins thus feature a significant content of hydrophobic amino acid side chains, especially LU, EL, VAL, P, and TRP. These are positioned so that they are clustered when the protein is folded, forming a hydrophobic protein core. The peptide bonds of proteins. Conformation of a peptide is defined by its dihedral angles or torsion angles called phi, psi, and omega. These are the angles for rotation about each of the 3 repeating bonds in the peptide backbone of the protein at the intersection of 2 planes. Figure 1.22. The important dihedral angles in a peptide are defined by the 3 bond vectors connecting the C, N, C bonds with the rotation occurring about the N, C bond and the N, C, N bonds. Rotations of the polypeptide backbone around the bonds between N, C, A are called F, and between C, A, C is called Y. The 6 atoms of the peptide group all lie in a single plane, with the oxygen atom of the cardinal group present in a transposition to the hydrogen atom of the amide nitrogen. Studies done by Collin and Corey revealed that the peptide C, N bonds cannot rotate freely because of their partial double bond character. Rotation is permitted about the N, C, and the C bonds. Because of the restricted rotation of these peptide bonds, the range of conformations possible for a polypeptide chain is limited. Figure 1.22 dihedral angles around peptide bond. Schematic diagram showing the various dihedral angles of amino acids. The restricted rotation of the peptide bonds around these dihedral or torsional angles is important for the protein conformation. Ramachandran plot. Since the peptide bond is planar, the dihedral angle W is usually restricted to 180 degrees trans or 0 degrees cis conformation. The distance between the adjacent CA carbons of 2 peptide bonds is 3.8 and 2.9 up respectively. Most of the peptide bonds, except proline, are present in trans configuration. In principle, the dihedral angles can have any value between -180 degrees to 180 degrees, but many values are restricted by steric interference between atoms in the polypeptide backbone and amino acid side chains. Allowed values can be visualized in a Ramachandran plot which depicts the allowed dihedral angle combinations by representing Y against F. Figure 1.23. This was introduced by Professor G. N. Ramachandran. Figure 1.23 Ramachandran plot. This is a plot of the torsional angles F and Y depicting the allowed regions for protein secondary structures. Ramachandran plot gives us a distribution of torsional angles in a protein structure. It serves as a map to visualize allowed and restricted angles for a stable protein conformation. It has been observed that the average F and Y values for A helices and B sheets are centered around -57 degrees, -47 degrees, and -80 degrees, plus 150 degrees, respectively. Protein folding. For a protein to function properly, it has to be able to fold into its native structure. The earliest protein folding studies were done by Christian Amundsen using RNase A, for which he was awarded the Nobel Prize in 1972. Proteins are held together by molecular interactions like the hydrophobic interactions and the disulfide bonds, and the thermodynamic stability of the folded protein structure. The thermodynamic stability of a protein helps find its most stable state, whereas the hydrophobic interactions within the protein stabilize the interactions within the secondary and tertiary structure of the protein. These hydrophobic interactions make the globular proteins acquire distinct compact conformations in water. Disulfide linkages also help in bringing the protein structure together by linking the different tertiary and quaternary structures together. Things can go haywire if the protein folding machinery is not in place. It can also lead to protein denaturation, i.e. complete loss of protein structure and function. It can also lead to various diseases. A classic example of a disease caused due to protein misfolding is Alzheimer's disease (AD). It is a neurological degenerative disease resulting in the formation of dense plaques of B-amyloid proteins in the brain. Every cell has a machinery to help in protein folding. These proteins are known as chaperones. The chaperone protein, heat shock protein 60 (HSP60), was first shown to be responsible for proteins refolding inside the mitochondria. Its bacterial equivalent is ROL. In an independent process, ROL, a cylindrical protein, takes up unfolded proteins into its core compartment. Figure 1.24. Once the unfolded protein chain enters inside, another protein called ROS functions to cap the ROL cylinder. During this step, a large-scale conformational rearrangement occurs in ROL that causes the hydrophobic amino acid residues to recede. This makes the ROL's interior relatively polar, somewhat like the cellular environment. This is essential as this makes up for a conducive environment for the protein to refold itself properly being ejected. Figure 1.24 ROL grows. A schematic diagram showing protein folding by ROL grows. This ATP-dependent protein folding process is essential for protein folding. Proteins as antibodies. Proteins which fight and defend the body against combat foreign substances are called antibodies. They are present in the G-globulin fraction of the serum. The G-globulin of normal blood serum has a mixture of various different G-globulins. If G-globulins are separated by gel filtration, the first fraction obtained has a molecular weight of 900,000. This fraction is the IgM class of antibodies. The next 2 fractions obtained are beta and antibodies. 2 polynucleotides helical chains each coiled around the same axis to form a right-handed double helix. A helix is right-handed if the helicity propagates in a clockwise direction when viewed along the axis of the helix from either end. 2. The 2 DNA strands are antiparallel to each other, which means that the 5' end of one strand is paired up with the 3' end of its complementary strand. So, if one DNA chain is in 5 feet right pointing arrow 3, along the positive z-axis, then the paired up chain must be in 3 feet right pointing arrow 5, along that axis. 3. The hydrophobic nucleophilic bases, purine and pyrimidine, are stacked together to form the core of the helix and the backbone of the polynucleotide chain, with alternating deoxyribosugar and phosphate group being hydrophilic remains outside of the helix facing the surrounding water. This position of phosphate groups also minimizes the repulsion among the negatively charged phosphate groups. 4. The nucleotide bases from each chain are stacked one after another, like stacked coins, and the planes of the bases are held perpendicular to the long axis. The bases from one chain form strong hydrogen bonding interactions with the paired bases in the same plane from the opposite chain. Watson and Crick found that the adenine A base of one chain always pairs by forming 2 hydrogen bonds with the thymine T base of the opposite chain, and the guanine G base from one chain always pairs by forming 3 hydrogen bonds with cytosine C base of the opposite chain. This base pairing provides a rationale to the target's rule. This specific base pairing of AT and GC, known as the Watson-Crick base pair, is shown in figure XX. Since G and C form stronger interaction, forms 3 hydrogen bonds, then A and T forms 2 hydrogen bonds. Separation of paired DNA strands becomes more difficult with a higher G as defined as C to equals T ratio. The distance between 2 base pairs in the same chain is 3.4a. The length of one turn of the double helix is 34a, so the helical structure repeats after 10 residues on each chain. The distance of the phosphorus atom from the fiber axis is 10a. 5. In DNA structure, the glycosidic bonds of a base pair are not diametrically opposite each other, and this causes the formation of 2 exterior grooves, major grooves and minor grooves in bDNA and those in between its sugar phosphate chain. The minor groove contains the pyrimidine O2 and the purine N1 of the base pair, and the major groove is on the opposite side of the pair. The major groove is wider 12a and deeper 8.5a than the minor groove, which is 6a wide and 7.5a deep. Figure 1.32. 3 different conformations of the DNA double helix. A-DNA is a short, wide, right-handed helix. B-DNA, the structure proposed by Watson and Crick, is the most common conformation in most living cells. C-Z-DNA, unlike on bDNA, is a left-handed helix. The different forms of DNA have several different structural features. Table 1.4. Table 1.4. The different forms of DNA. Unusual forms of DNA. bDNA is the most common conformation found in most of the living cells. However, other 2 conformations of biologically active DNA are also possible. A-DNA, a shorter and more compact right-handed helical structure of DNA whose base pairs are not perpendicular to the helix axis is in bDNA. A form is mostly favored in solution, which is relatively devoid of water. The reagents used for the crystallization of DNA generally tend to dehydrate DNA. That is why short DNA molecules tend to crystallize mostly in A from. Another type is Z-DNA, a left-handed double helical DNA. The reverse circular dichroism spectrum of such DNA is interpreted as the left-handed winding of the DNA chains. Since the discovery in the crystal structure of Z-DNA in 1979, several interesting facts about this type of DNA have been established, and it has been found that Z-DNA is associated with both Alzheimer's disease and systemic lupus erythematosus. The transformation pathway of bDNA to Z-DNA has been experimentally proved using SM-FRET assay, and a major portion of those disease-affected DNA was found in BZ intermediate conformation. Figure 1.33 Structural variation of nucleotides in DNA. A. Sterically permitted. A. Sterically permitted anti and syn conformation of purine bases with respect to the attached ribose unit. B. The possible rotation of 7 different bonds are shown in arrows. Unusual structure of DNA can occur due to the presence of certain sequences also. For example, in one DNA chain, presence of adenosine nucleotide in succession results bent in the DNA helix, and the presence of 6 adenosine serially produces a bend of almost 18 degrees. Other most commonly appeared structural abnormalities in DNA helix are listed below. 1. Hairpin loop occurs when a single DNA chain contains complementary nucleotide bases in the correct sequence to those that appeared a few bases earlier in the same strand. The presence of these types of base sequences causes complementary base pairing in a single chain, intrastrand base pairing, by folding back on itself and resulting in some of the bases unpaired inside the loop. For example, if a DNA strand contains sequence AGCCT followed by some other base sequences, including AGGCT, then the strand is capable of creating a hairpin loop. In a DNA double helix, when an inverted repeat of base sequence occurs, having twofold symmetry over two. In a DNA double helix, when an inverted repeat of base sequence occurs, having twofold symmetry over 2 strands of DNA, these types of sequences are known as palindromes, and both strands of duplex DNA forms hairpin loop. Then those are called cruciform structure. Figure 1.34. Figure 1.34 hairpin and cruciform structure of DNA. Nucleotides in the Watson-Crick base pair to form additional hydrogen bonds results to the pairing of the third polynucleotide chain to the existing double helix, and this non-Watson-Crick pairing is called Hooke's pairing, where the N7 position of the purine base is a hydrogen bond acceptor, and C6 amino group as a donor forms a hydrogen bond with the Watson-Crick N3C4 base of the pyrimidine base. Figure 1.35. The presence of palindromic sequence can cause interstrand base pairing. A. Intrastrand base pairing in a single strand is called hairpin. B. If both strands of a duplex DNA are involved, it is called cruciform. 2. Triplex DNA is a double-stranded DNA with one additional oligonucleotide chain that forms a hydrogen bonds with the bases that are already included in the double helix strands of DNA. The ability of the Figure 1.35 Watson and Hooke's base pairing of DNA. The picture displays 2 different natures of bonding among the nucleotides. 3. Hinge DNA or HDNA is also a triple helix structure that exists based on the Hooke's pairing base composed of triple-stranded and single-stranded regions. These are found in polypyrimidine or polystyrene tracks. For example, repeating copolymers of DTDCN. DGN sequences TC. AGN can assume a hinge DNA structure HDNA. 4. G-tetraplex are the quadruple-stranded structures of DNA formed in guanine-rich sequences of nucleic acids. In this form, 4 guanidines centered around a monovalent cation to form a square arrangement which is stabilized by Hooke's pairing. In comparison to the typical DNA structure with 2 grooves, this 4-stranded structure has 4 grooves providing a variety of surfaces for interactions with ligands. DNA denaturation and renaturation. DNA denaturation is a process in which the native structure of DNA collapses, and its 2 complementary strands become separated to take up a flexible and rapidly fluctuating conformational state known as the random coil. The separation of the 2 strands occurs abruptly at a certain temperature, and the process is called the melting of DNA. The temperature at which 50% of the helical structure of DNA is melted is known as the melting temperature of DNA. The 2 strands in DNA are held together by hydrogen bonds, stacking interactions with hydrophobic effect between complementary base pairs in each strand. By applying heat or by addition of denaturant, or by increasing the pH level, those interactions can be disrupted to form relatively freely jointed single DNA strands. The G is defined as C base pair in DNA forms 3 hydrogen bonds, while as defined as T base pair forms 2 hydrogen bonding. More energy is required to separate the G is defined as C base pair than the A is defined as T base pair, and the melting temperature of DNA increases with the content of the G is defined as C base pair. Denaturation of DNA can take place by temperature, heating the DNA solution above A. Figure 1.36 Plot of DNA denaturation kinetics. The midpoint of the curve, melting temperature Tm, is the temperature at which half the DNA population gets denatured. Relationship between base composition and DNA stability. High GC amounts possess a higher Tm than DNA with a higher AT content, keeping other parameters the same. At certain temperatures, adding chemical denaturation agents like urea and formaldehyde, these chemical reagents increase the aqueous solubility of purine and pyrimidine groups and changing the pH of DNA solution at an acidic and alkaline solution in which ionic changes of purines and pyrimidine bases can occur. Denaturation at one part of DNA destabilizes the remainder resulting in a narrow temperature range during melting, which indicates DNA melting is a cooperative process. Melting of DNA does not break any covalent bond in DNA. One important point in this aspect is that inside the cell, DNA is not melted by the addition of heat. Instead, a specific enzyme called helicase is required, which supplies chemical energy in the form of ATP to disrupt the double helix. After denaturation, following properties of DNA are changed. 1. Increase in absorption wavelength. Native DNA has a maximum absorbance at 260 nanometers. Upon denaturation, the absorbance at 260 nanometers increases by approximately 40%, i.e. hyperchromatism is observed in the UV absorption spectrum of DNA, generally attributed to the unstacking of the DNA base pairs. Because of this property, DNA denaturation can be followed by monitoring absorbance at 260 nanometers. The amount of nucleic acid present in a solution can be measured by applying Beer-Lambert law. A equals log I O equals epsilon CL. A is the absorbance of the solute, or optical density. I0 is the intensity of incident light at a given wavelength L. I is its transmitted intensity at L. Epsilon is the molar extinction coefficient of the solute at L. C is its molar concentration, and L is the length of the light path in centimeters. The value of epsilon varies with L, and the plot of epsilon versus L for a solute is called the absorbance spectrum of the solute. 2. Decrease in specific optical rotation. Native double-stranded DNA has positive specific rotation, which decreases in a significant amount after the melting of DNA. 3. Decrease in viscosity. Owing to the rigid, double helical, rod-like character of DNA, it possesses a high viscosity, and after denaturation, as the strands are separated, the viscosity decreases. Hypochromic effect of DNA. The term hypochromicity is used when the absorbance of material is decreased at a given wavelength of light. DNA can be renatured if the solution of denatured DNA is cooled rapidly, much below the melting temperature. This process of renaturing of DNA is called the annealing of DNA. Since in the double-stranded DNA, the nucleotide base pairs are stacked with each other, the absorbance due to absorption of aromatic chromophores in nucleotide bases mainly decreases in comparison to the single-stranded form of that DNA molecule. This phenomenon is known as the hypochromic effect of DNA. DNA renaturation kinetics. The rate at which each denatured DNA sequences will be completely renatured depends on DNA concentration, reassociation temperature, cation concentration, and viscosity. The rate of renaturation follows the kinetics of second order in DNA phosphate concentration. Rate of renaturation increases by lowering the temperature below Tm and reaches a broad flat maximum from 15 degrees to 30 degrees Celsius below Tm and then decreases by the decrease in temperature. For a given DNA, a fixed DNA phosphate concentration rate of renaturation decreases in a decrease in molecular weight. The rate of renaturation depends on the ionic strength of the solution below 0.4 m for an electrolyte e.g. NaCl and is almost independent of salt concentration above this ionic strength. C0T value. In DNA, very often, patterns of nucleic acids occur in multiple copies throughout the whole genome. Those are called repeated sequences or repetitive elements and are responsible for rapid reassociation kinetics of single-stranded DNA. In many organisms, such as human organisms, a significant fraction of the sequences consists of repetitive DNA. There are different classes of repeated sequences like tandem repeats, interspersed repeats, etc., and some leads to the increasing manifestation of diseases. Based on the principle of DNA renaturation kinetics, in 1960s, Roy Brin and his colleagues developed a technique to analyze the C0T value. Based on the principle of DNA renaturation kinetics, in 1960s, Roy Brin and his colleagues developed a technique to analyze the C0T value, which is a measure of how many repetitive DNA sequences are there in a DNA sample by measuring the rate of reassociation of complementary strands of DNA derived from the same source. In this study, double-stranded DNA is shredded into pieces with almost several hundred of base pairs and denatured to single-stranded DNA by heating. By lowering the temperature subsequently, the rate of renaturation or annealing is monitored. Renaturation kinetics of denatured DNA is used as a very important tool for measuring the genome complexity, total length of different sequences of DNA. DNA reassociation follows second-order kinetics and is described by C0T 1/2. During denaturation, high molecular weight DNA is broken down into pieces of approximate lengths. Renaturation rate of the melted molecule may be presented by the equation as dC/dt equals minus Kc2, 1. In equation 1, K represents the second-order rate constant, and C stands for the concentration of melted DNA strands with respect to time t. When two single strands begin to pair with each other, there is a reduction in the rate of C in the mathematical expression of second-order kinetics. At t equals 0, the initial concentration of the starting entirely separated molecule is C0. At a particular given time t, the concentration of residual melted DNA is given by the following equation. C0 equals 1, 1 plus Kc0t, 2. T1/2 that is the time required for 50% reassociation of DNA, then C0 equals 0.5. So, 0.5 equals 1, 1 plus Kc0t 1/2. Or, 1 plus Kc0t 1/2 equals 2, i.e. C0t 1/2 equals 1K. The product of C0 and T 1/2 is known as C0t 1/2. C0t 1/2 has an inverse relationship with the rate constant. As it is specified by the initial concentration and required half time, a higher C0t 1/2 value corresponds to relatively slow renaturation. A graph of the fraction of single-stranded DNA reannealed (CO) as a function of C0t on a semi-logarithmic plot is referred to as a C0t curve and represents the reassociation kinetics of duplex DNA. The rate of renaturation is presented in the plot with the percentage of single-stranded DNA versus log C0t value, where C0 is the initial concentration of ssDNA in moles, L and T is the renaturation time in seconds. Sometimes, a buffer factor is multiplied with C0t to account for the effect of cation in the speed of renaturation. DNAs with highly repetitive sequences reanneal at low C0t values 10 - 4 to 10 to 1. Moderately repetitive DNAs reanneal between C0t values 10 0 to 10 to, and DNAs that reanneal at higher C0t values are almost non-repetitive. Figure 1.37 plot of DNA renaturation kinetics. A. The C0t plots of small-sized DNA. B. Determination of repetitive sequence in DNA. C value paradox. C value is the amount of DNA in picogram present within a haploid nucleus or only half the amount in a diploid somatic cell of a eukaryotic organism. In 1948, it was reported by two scientists that the amount of nuclear DNA content is constant in all the cells in all the individuals within a given animal species, and the concept of this proposal was that DNA is the substance of which genes are made. One might expect a correlation between the genome size, C values, and the complexity of the organisms. It was soon found that there can be a large difference in C values of very similar organisms, and C values cannot be directly correlated with the gene number, which is a reflection of the complexity of an organism. On the other hand, genetic information is encoded by DNA, so C values were assumed to be constant, although they did not have any relationship with the resumed gene number. This paradoxical concept was described as the C value paradox in 1971 by C. A. Thomas Jr. However, after the discovery of non-coding DNA, the C value paradox was solved. In the eukaryotic cell, most of the DNA is non-coding, which supports the fact that genomic sizes reflect the gene number in an organism. Genome complexity. Eukaryotic genomes are larger and more complex than that of prokaryotic genomes. However, from the C value paradox, we came to know that the genomic size of most of the eukaryotes are not directly related to the genomic complexity. This is because a large number of eukaryotic genes consist segments of coding sequences, exons, which are interrupted by many of the non-coding sequences, introns. Introns are deleted from the genes before it is encoded to protein in a process called RNA splicing. Another major contributor to the large size of the eukaryotic genome is the multiple copies of genes, called gene families. Sometimes to produce a large number of a certain protein, multiple repetitions of genes are required, or gene families may be transcribed in different stages of the development of the organism. In some cases, mutation of gene copies results into a loss of their ability to produce functional biological molecules and become inactivated. Such non-functional gene copies are called pseudogenes, which do not have any functional contribution, but they increase the structural size of the genome. The presence of repetitive DNA sequences also increases the complexity of the genome in eukaryotic cells. DNA supercoiling. Most of viruses and bacteria have circular DNA as seen by an electron microscope. The twisted appearance in the tertiary structure of some of the circular DNA is known as DNA supercoiling. The antiparallel strands of DNA are covalently joined to form the circular duplex molecular structure. In this type of structure, it is not possible to alter the number of times one strand wraps about the other without cleaving at least one of its nucleotides. This phenomenon was mathematically expressed by James White in 1969 as linking number L equals twist T plus writing number W. Linking number is the number of times one DNA strand wins about the other. Twist is the number of complete revolutions of one polynucleotide chain around the duplex axis in the particular conformation under consideration. Writing number is the number of turns made by the duplex strand about the superhelix axis in the conformation of interest. Supercoiled DNA can be relaxed by treating with one endonuclease enzyme, pancreatic DNase I, which cleaves one strand of duplex DNA. DNA intercalating agents can control DNA supercoiling by unwinding DNA. Supercoiling of DNA is very important because it helps in the packaging of the long DNA molecules almost thousands of times that of the cell length into the cellular nucleus for eukaryotes. RNA structure. Ribonucleic acid RNA is a polymer of ribonucleotides and has quite a similar ribonucleic acid RNA is a polymer of ribonucleotides and has quite a similar basic structure to DNA. In contrast to typically long and double-stranded DNA, RNA is much shorter and usually single-stranded. DNA sugar phosphate backbone is made up of deoxyribose. 2' hydroxyl group is absent, but in the RNA sugar phosphate backbone, ribose sugar, the pentose sugar, is present. RNA nucleotide uses 4 nitrogenous bases, purine base A9A, guanine G, and the pyrimidine bases cytosine C and uracil U. A variety of forms of RNA are available, and those are messenger RNA, mRNA, transfer RNA, ternate ribosomal RNA, rRNA, non-coding RNA, and small nuclear RNA, snRNA. Each form of RNA has different function and activities in the biological system. Messenger RNA. Messenger RNA is the intermediary between DNA and its protein products. mRNA copies a section of DNA and serves as a template for the synthesis of proteins. mRNA carries the genetic messages from DNA to ribosome, where protein is synthesized according to the instruction carried by mRNA. This type of RNA consists mainly of coding sequences along with untranslated sequences and poly A tail. Codons consist of 3 base pairs which specifically encode for specific amino acids. Coding sequence or coding region in mRNA begins with a start codon, the first codon to be translated by the ribosome and ends with a stop codon.